Hey everyone, welcome back. Ever tried figuring out like what a single gene actually does? Like this massive instruction manual, right? For building a whole organism.
Yeah.
But every single gene, it's like it's written in some kind of secret code.
Yeah.
So today we're going to dive into, well, a technique that cracks that code. Functional Complementation Analysis, or FCA for short. We're using a 2016 article from the Journal of Visualized Experiments as our guide.
It's a really good one, too. The authors, that's Hudson, Harkness, and Safka, they don't just explain FCA. They walk us through these really fascinating case studies, show it in action.
Nice. So it's like our detective's handbook for this deep dive then.
Exactly.
Okay, so how does FCA work? Imagine like a machine with a missing part. Like,
Yeah.
say your blender just gave up, it just won't blend.
Right, completely busted because of that one missing piece. Now imagine you've got this box of spare parts, right? But they're from a totally different blender, maybe even a different brand. FCA is kind of like testing out those random parts one by one just to see if any of them can fix your poor blender.
And if that replacement part does get it working again, then we know it was probably doing a similar job in the original machine. But we're talking about genes, not blenders, right?
Exactly. Scientists use FCA to study, well, mutant organisms.
Hmm.
These are organisms where a specific gene, it's not working right. And then they'll take a similar gene, we often call it a homologous gene, from a different organism, and they introduce it into that mutant.
So like giving our busted blender apart from like a food processor or something sounds crazy.
Sometimes it actually works.
No way, really.
Yeah. And when it does work, when that new gene restores the function that was lost, it tells us something huge, that the introduced gene is probably responsible for that same function in its original organism.
Wow, that's incredible. So it's like solving this biological jigsaw puzzle, but using pieces from all these different puzzles.
Yeah.
Okay, enough analogies for now. Our article, it highlights how researchers used FCA to solve some real biological mysteries. And the first one, it involves lysine,
Lysine, yeah.
which is an essential amino acid, right?
It's crucial for us humans, absolutely essential for building proteins. Our bodies, though, we can't make it ourselves. Got to get it from our diet.
Which is where plants come in. They've got that lysine-making process down, I'm guessing.
Oh, they do. But here's where it gets interesting. See, the way plants produce lysine, completely different from how bacteria do it.
Wait, really?
Oh, yeah. It's like two completely different factories making the exact same product.
No way. Two totally different paths through evolution, and they both ended up with the same amino acid? That's wild.
Yeah, and FCA, that was the key to figuring this whole thing out. Researchers used it to pinpoint this one specific gene, it's called dapl, and it has a crucial role in how plants make their lysine.
Okay, so they took this plant gene, this dapl, and they put it into a bacterium that couldn't make its own lysine. What happened?
Well, that bacterium, it suddenly started producing lysine. It was pretty clear evidence that this dapl gene, it was the missing puzzle piece, the gene responsible for that key step in how plants do it.
That's amazing. But why should we care about how plants make lysine? It's not like we're, harvesting it from dandelions in the backyard.
True, but this discovery, it could change things big time. For agriculture, food security, the whole picture. Imagine like being able to boost the lysine content of crops. That could be a game changer, especially in areas where, lysine deficiency is a real problem.
So this tiny discovery, figuring out just one gene, could actually impact people's lives in a huge way. That's really something. Okay, so we've seen FCA at work figuring out plant lysine production, but our deep dive isn't over yet, is it?
So from plants, let's jump to bacteria, specifically how they defend themselves. It's like, imagine a bacterium, right? But it's this tiny little knight all suited up in armor.
Okay, a tiny armored knight. I like it. What's the armor made of?
That armor, it's called peptidoglycan, and it basically gives the bacteria its structure, its strength.
So all bacteria have this peptidoglycan armor. But I'm guessing it's not, like one size fits all.
You got it. There are some key differences in that peptidoglycan layer. We're talking gram-negative versus gram-positive bacteria. And here's why it matters a lot. Of plant pathogens, the bacteria that cause diseases in plants, they're mostly those gram-negative types.
So if we can understand how those gram-negative bacteria build their armor, maybe we can protect our food supply a bit better.
Exactly. And guess what? FCA comes in clutch again. Researchers used it to find this gene called mure. It's essential for building that bacterial armor, especially in those gram-negative baddies.
This makes me think about the whole antibiotic resistance problem, Could this discovery, this mure thing, could it help us make new antibiotics? Ones that target this armor building process?
$1 million question right there. But the more we know about these essential processes in bacteria, how they work, the better we can target them, even as they evolve and get resistant to our current weapons.
It's like a constant arms race then. Scientists versus the bacteria. Okay, so we've seen how bacteria suit up, but what about other threats? What do they do when they're low on food, for example?
Well, bacteria have this pretty fascinating trick. It involves slime.
Slime. Now you've really got my attention.
Okay, so when bacteria get stressed out, like if they're running low on nutrients, they activate this thing called the stringent response. It's kind of like their version of hunkering down, riding out the storm.
Makes sense, a very bacterial thing to do.
Right.
But where does the slime fit in?
So there's this one bacterium, Novosfingobium sp, and it's got this very unique way of dealing with stress. It gets super slimy, almost like it's building a slimy little fortress around itself for protection.
Now that's what I call a sticky situation. But seriously, how'd the researchers figure out which gene was responsible for all that slime? Don't tell me it was FCA again.
You know it. Using SCA, they pinpointed the gene, it's called Archish, and it controls the production of that stress-induced slime.
Okay, gotta ask, are there actually uses for bacteria slime? This is where you blow my mind again, right?
Well, think about biofilms. You know those slimy communities bacteria form? They can stick to pretty much anything. You've probably seen them, even in your own home. Like that stubborn gunk that builds up in your shower. Biofilm central.
Okay, I'll admit, I've done battle with some shower slime in my day. But how's that related back to this ERS gene and our slimy Novosfingobium?
It turns out, Artesh, pretty crucial for how Novosyngobium sp forms biofilms. And it's not just a nuisance in your bathroom. Biofilms are a big deal in hospitals. They can form on medical devices, lead to infections, and in industrial settings, forget it. They can clog pipes, contaminate equipment. It's a mess.
So if we could, like, control that slime production, maybe we could stop those biofilms from even forming.
That's the idea. So this discovery, figuring out this ERS gene and how it affects Lyme, it gives us a whole new way to think about controlling biofilms. It's like this perfect example of how understanding a tiny little detail about this microscopic organism can have huge implications.
This is why I love science. Just when you think you've got it figured out, there's bacteria building slime fortresses. But we're not done yet. What else did these researchers uncover?
So besides lysine, there are other amino acids that are essential, for us humans. We can't make them ourselves, have to get them from our diet. And just like with lysine, plants and bacteria have found their own ways of making these essential building blocks.
Okay, you've got my attention. Which amino acids are we talking about now?
This time, it's tyrosine and phenylalanine. And wouldn't you know it, researchers used FCA to highlight just how different the production methods are between plants and bacteria.
So multiple factories in nature for these amino acids, too.
You got it. Plants and bacteria, totally different worlds in a lot of ways, but they've both evolved their own really ingenious ways of making these same essential molecules.
Walk me through it. How was FCA used to show these different pathways?
So they took a gene from that model plant, Arabidopsis thaliana. This gene, it's called AT5G36160, and it's involved in making tyrosine and phenylalanine in plants.
Okay, so they took this plant gene, and I'm guessing popped it into a bacterium that couldn't make its own tyrosine or phenylalanine, kind of like that lysine experiment we talked about.
You got it. They used E. coli for this one, and this particular E. coli, its tyrosine and phenylalanine production line was totally busted, couldn't make them on its own. But when they stuck that plant gene in,
It worked. The E. coli, it started making its own tyrosine and phenylalanine using instructions from a plant.
it did, which proved that even though they come from totally different kingdoms, that plant gene, it could function in a bacterium. It just goes to show you, the shared evolutionary history of everything. Plants, bacteria, they split millions of years ago, but there's still these fundamental similarities in how they do these essential processes.
That's incredible. FCA can uncover these hidden connections between organisms. Connections we never would guess were there. Like
Yeah.
finding out you've got something in common with some distant cousin, but your families have been living totally separate lives for generations.
Haha, I like that. And it shows why FCA is such a versatile tool. And it's not just for bacteria either. Scientists are using it to explore genes in all kinds of organisms, plants, yeast, even mammalian cells.
Wow. So we've gone from blending up lysine to bacteria and slime armor. What other mysteries can FCA help solve, you think?
Well, think about it. There are still tons of genes out there. And we have no idea what they do. Just waiting for someone to come along with the right tool to unlock them. And FCA, it's that tool, like a master key for the whole tree of life.
It's pretty amazing to think about what's coming next in genetic research, especially with tools like FCA.
It really is. And, you know, the practical applications of these discoveries, how will this change things for us? New medicines, more resilient crops. Could we clean up pollution using microorganisms? The possibilities are huge.
Couldn't agree more. And on that note, we'll leave you to ponder all those possibilities, the amazing world of the microscopic, and the huge discoveries waiting to be made. This has been The Deep Dive. Catch you next time.